Well, I'm happy to introduce Dr. Alvin Huang, who's speaking to us today about stem cells in focus, the role of glial cells in a potential treatment for Angelman syndrome. He comes to us from Brown, and we are very happy to have you. Welcome. Forward, back, and laser. You want this or you want? That's good.
All right, I am exceedingly delighted to be here with you today, extending a warm welcome to families, researchers, and supporters gathered here on site or online. So my name is Alvin Huang. I am a neurologist turned neurobiologist, and my research journey has taken me through the intricate pathway inside the brain, exploring neurocognitive disorders across the spectrum of brain development and aging. So recently, my focus has shifted toward conditions that we all hold very dear to our heart, Angelman syndrome. And then my talk title is Stem Cell in Focus, the role of Glia cells in potential treatment for Angelman syndrome. Quite a mouthful indeed. So we will tackle this in 3 digestible parts. So the first part, we will talk about glia and a special cell type called oligodendrocyte. So what exactly are they and what role they play in the brain, particularly concerning Angelman syndrome. The second part is about human-induced pluripotent stem cell, a game-changing tool that comes with human-specific features not available in rodent models, and thus offer a revolutionary way for us to study the disease and test new drugs in a lab setting. And then for the third part, we will discuss our research that is generously funded by the FAST. So our team is leveraging our expertise in stem cell technology to dissect the Angelman syndrome disease mechanism in oligodendrocytes, aiming at uncovering new therapeutic targets. All right, Glia. So most of us know that when we learn something or notice things around us, this information is passed through the brain like electricity traveling through wires. So let's now imagine the brain as a sophisticated electric circuit board. So on this board, the neuron processes or nerve fiber acts like the intricate network of wires that allow electricity, or in our brain's case, information, to flow one component to another. Now, on an electric circuit board, it's not just the wires that matter. So, as you know, they are resistors, capacitors, as well as other components that control and modulate the electricity's flow, ensuring the board works efficiently and as intended. And in our brains, glial cells, they come in different shapes and sizes, play a similar role. So these glial cells, they aren't just passive supporters. They actively modulate the flow of signals in the brain, much like the components on a circuit board. So they help maintain the balance, provide the nutrients, and in some cases, even speed up the transmission of signals. So today, we are focusing on special kind of glial cells called oligodendrocyte, marked in color purple here. So these cells are like the electric tap of the brain. So they wrap around the wires or nerve fibers and help keep the electricity from leaking or getting lost or moving too slowly in the brain. So they make sure everything is wired properly and nicely connected through the synapses. And it's important for our brains to do amazing things, like to help us learn, remember, and even feel emotions. So this oligodendrocyte, they start off as little oligodendrocyte precursor cell, or OPC for sure. So let's just call them the whiz kids of the brain cell world. So they can sense and respond to the electric or the information signals passing around, and then that can multiply and transform into bigger and more specialized cells. And over time, these cells grow up to become the full-fledged oligodendrocyte, sending out processes to wrap nerve fibers in many fatty layers called myelin. So sort of like the layering electric tap to protect a wire, so which indeed pretty much look like onion skin under a high-resolution microscope. So this myelin not just insulates, but also speeds up the messages that our brain send. So it's a bit like upgrading to a faster internet cables, so allowing us to think quickly and efficiently. So if something goes wrong in this growth process, it can mess with our brain's electric system, and this might lead to slower learning or even cause our brain's electric signal to go haywire, resulting in seizures. And so these well-wrapped nerve fibers create what we call white matter in the brain, which we can actually see on brain MRI scan as the bright subway system of the brain lying beneath the gray matters, the less insulated outer layer of the brain that handles all the processing. And interestingly, this myelination process by oligodendrocyte is the very final touch to building our brain. So even though our neurons, they are already lined up and ready for action early on in a baby's brain inside the womb. So this myelination takes its time. Our brains keep fine-tuning this myelination well into our use. So in fact, it isn't until we are in our mid-20s that this process is fully complete. So that's why we keep getting smarter and better at solving problems as we grow from teenagers into young adults. But why this is important for Angelman syndrome? So multiple studies using brain MRI scans have shown that young people with Angelman syndrome, so ranging from babies, toddlers, children, teenagers, or even young adults, experience a further slowdown in the way their brains become insulated with myelin. So this is like the brain's wiring taking longer to get its protective coating, and which can affect learning and behaviors. But the good news is that it looks like they do catch up when they become adults. But since the period of childhood to young adulthood is super important for developing intellectually, so this delay can make learning tougher for these kids during those years. And then there's also something interesting going on at the level of genes. So we have already learned the unique genetics of Angelman syndrome and then the amazing functions in neurons in the beautiful talks before me. But it turned out that the UBE3A is also expressed in human oligodendrocytes. And so this report has made our research team curious about whether UBE3A has a job in helping oligodendrocytes grow and work correctly in our brain. And so to get around the challenges of looking at the human oligodendrocyte closely, because we can't just pick inside a living brain yet, so the researchers, including several scientists within our FAST community, others turning to an innovative approach using stem cells. And these cutting-edge techniques allow us to explore the oligodendrocytes in the lab setting, creating a kind of humanized models that can give us vital clues and help us discover new drugs. And these groundbreaking methods use what's called induced pluripotent stem cell, or iPSCs for short. So now, before we dive deeper, I'm going to walk you through some stem cell basics. So these indeed are the same materials I use to teach my course at Brown University, and I will make it just as clear for you today. So in simple terms, stem cells like the starter kits of life. So the totipotent stem cells are the first cells from fertilized eggs. So they are like all-in-one tools, able to create a whole baby and the life support system like the placenta. And pluripotent stem cells come next. They are like specialized kits. They can make any part of the baby, but not the support system. So lastly, multipotent stem cells are like the final kits. They can only make certain parts, like cells of the heart, brain, or blood. And then the oligodendrocyte precursor cell, or OPCs, we talked about before, are a type of these cells with a set path to become a specific brain cell type, oligodendrocyte. So the development of cells in the bodies was long thought to be strictly one-way journey, or like a marble rolling downhill, going from a do-it-all cell state to more specialized ones without turning back. So this indeed is, the most famous analog used in the textbook. Because just as a marble won't roll back up a hill and against the law of gravity, it was long believed that the cells couldn't reverse their specialization for decades, if not for centuries. But Dr. Shinya Yamanaka's groundbreaking work showed that a mature cell can indeed be reprogrammed or induced back to an all-purpose cellular state, much like dodging the marble back up the slope. So this discovery made in 2006, and which won a Nobel Prize within just 6 years in 2012, introduced a new kind of versatile cells called induced pluripotent stem cells, originally derived from a mature skin cell type fibroblasts. And then today, we can use skin or blood samples from people with genetic condition like Angelman syndrome, and we can make the stem cells, they have the same genetic makeup as the donors. And so this led us to create the exact type of cells affected by the disorders in the dish, and making it easier to study the disease closely. Plus, this method is great for quickly testing lots of different drugs to find the best treatment. So my lab has been working hard on creating or improving these iPSC-based technologies. So we either use the stem cells given by donors or using the CRISPR techniques you've heard a lot today to introduce or create specific changes, like those stop the UBE3A gene from working. And then we instruct these cells to grow into neurons, glia, or other brain cells. We can also grow them in complex 3D structures, like the organoids we heard about earlier, that sometimes we dub as mini brains. So we are also spreading the word about the techniques we develop, and we published the exact experimental procedures in great details to promote the utility, and like in this book we just published this summer. And now we've learned a lot about oligodendrocytes and iPSCs. Let's talk about our research project. So in which we used oligodendrocyte made from the iPSCs to find out how Angelman syndrome happens, and even better, to discover new drugs that might treat it. We have developed a really efficient way to create a human oligodendrocyte from iPSCs, where we can mimic the nature development, but much faster, in weeks instead of in years. So we start with OPC, oligodendrocyte precursor cells, let them grow, and then they mature into functional oligodendrocyte. They can form myelin. So we can actually watch the myelination happens, seeing the oligodendrocyte part shown in red here, wrapping around the nerve fiber, shown in green here. And thanks to these methods, our fantastic postdoc scholar, Dr. Xin Yang, together with other of our lab members, have discovered that UBE3A indeed is active in the early stage of oligodendrocyte development and actually helps these cells grow and mature properly. In the case of Angelman syndrome, gene mutations in our donor iPSC cells that we acquire from Dr. Jiang can indeed deactivate UBE3A, and we think this is why young people with Angelman syndrome experience slower myelination process. So our team is thrilled about this because we know, we found a new way that can help us do something with this. So we have been using such stem cell-based methods to screen a collection of 50 plus different drugs, checking to see if any could counteract the issues caused by Angelman syndrome that stop oligodendrocyte from growing properly. So as shown here in the graph here, so there's a roughly 50% reduction in the growth of OPCs. And then, so I also would like to point out that so these drugs, they are selected for their own ability to possibly correct the genetic issues or protect brain health or encourage the growth of myelin to help signaling transmission. And then we also prioritize the safety profile, as many of them actually has already gone through the clinical trial for non-relevant brain disorders. And our findings are very surprisingly and also very exciting. So a handful of this drug actually restore normal growth to these struggling Angelman syndrome OPCs. And interestingly, this effective drug, they interact with parts of cells in ways that we didn't expect. So revealing new insights into how Angelman syndrome might be treated. So we are now deeply involved in figuring out the science behind why these drugs are working and believe we are on the verge of understanding a new aspect of the disease. And then, so the most exciting results came when we picked one drug that stood out in our tests and give it to mice that have Angelman syndrome. So we are amazed to see the significant improvements in the ways these mice move and learn, and pretty much like the behavior assays you have all learned in Allison's work, but we plotted here, and then we are just so pleased to see the exchanges made by the drug treatments, going up and down to bring back the normal conditions. And we believe this can mirror the potential benefits they have for the humans with these conditions. So this led us to hope that we might be on the path to developing a treatment that could truly make a difference for those living with Angelman syndrome. All right, so in summary, I hope my talk conveys the message well enough to let you appreciate the function of oligodendrocyte as well as the contributions to disease development. And I also would like to really, promote the utility of the human stem cell-based systems. And to us, these projects opens up a fresh avenues that focus on correcting glia dysfunctions that we believe can complement the groundbreaking works in restoring neuron UBE3A expression. Okay, I'd like to give a huge thank you to our incredibly wonderful research team. And unfortunately, this is not the most updated pictures we have. And I also would like to shout out to our collaborators inside or outside of Brown, and the Drs. Mayoral John Marshall, as well as Dr. John at the Yale. And in fact, soon you will be here about another exciting work at the Brown done by Dr. Marshall. And of course, I would like to thank again for the generous support of the FAST. They really make this work to progress much faster than what we originally expected. And then, the community gathered here today is truly second to none in its commitment to fostering significant scientific breakthrough for better treatment.